Hello, everyone. Thank you so much for joining our Multiplex IF workshop today. My name is Afrida Rahman Enyart. I am a scientific liaison here at Protein Tech. And before I go ahead and introduce our speaker for today, I wanted to share a couple company slides so that you know who we are as Protein Tech. So Protein Tech is a leading manufacturer of antibodies and immunoassays. We have antibodies against over 13,000 targets. We also sell ELISA kits, IHC kits, and cell separation kits. Within our Protein Tech brand, we also have our Humankind line, and Humankind manufactures cytokines and growth factors. And all our cytokines and growth factors are expressed in human HEC293 cells, meaning that they have native folding, post-translational modifications, and glycosylation. We also offer both RUO and GMP grade products. The third line that we have within our Protein Tech brand is our Chromotech line. And Chromotech manufactures nanobody-based reagents that are ideal for immunoprecipitation, immunofluorescence, and protein purification. What makes Protein Tech different from other companies is that we manufacture our products in-house and sell directly to the customer. We're also very transparent about our product performance and include original validation data and quality control. So with that, I want to introduce our speaker for today's workshop, Derek Sung. Derek is an MD PhD student at the University of Pennsylvania studying mechanisms of vascular development in the embryo and placenta. He actually recently defended his PhD, so now he has Dr. Derek Sung. He utilizes both in vitro and in vivo models in his projects. He also specializes in immunofluorescence and fluorescence microscopy, and he sees these as powerful tools to answer all types of scientific questions. So with that, I will hand it over to Derek, and thank you, Derek, for being here today and for running this workshop for us.
Great. Thank you so much, Afrida. Thanks for Protein Tech for having me. I'm really excited today to be talking to all of you about my favorite scientific technique ever, immunofluorescence. So Afrida gave me a wonderful introduction. I'm just going to talk a little bit more about my path and some of the sort of how I got into imaging and some of the different types of imaging that I've done. So I did undergrad at Cornell University in Ithaca, New York. I graduated in 2016 with a degree in nutrition. And at Cornell, I was doing a lot of research into mechanisms of calcific aortic valve disease, so heart disease. And we did a lot of mouse models and in vitro cell culture models. And that was kind of my start to imaging and starting to do some confocal imaging. After graduating, I worked for a year at the National Institutes of Health in the National Heart, Lung, and Blood Institute with Bob Adelstein studying non-muscle myosin 2. And there I really focused a lot more on cell biology. I did a lot of work with the cytoskeleton, a lot of super resolution microscopy, and I also kind of ventured outside my typical scientific field and I studied testes development. And then in 2017, I started my medical scientist training program, which is my MD PhD program at the University of Pennsylvania. I did medical school for 2 years before starting graduate school. I actually just finished, as Afrida said, and defended my thesis just this past Monday, thankfully successfully. And I was studying vascular development using mouse models and a lot of embryonic developmental mechanisms of developmental angiogenesis. And in September, I'll be heading back to medical school. But here I was focusing a lot on confocal microscopy. And I'm sure a lot of you probably, or not a lot of you, some of you might know me from my kind of like online personality, which is my Instagram account called Immunofluorescence. And today we're mostly going to be talking about immunofluorescence staining, like the protocol. But of course, there's much more to fluorescence microscopy than just the staining itself. A question I often get is like, how do I analyze my images and how do I get data from these images that I've taken? And I just want to point to a potential resource for some of you. I've created some of these tutorials, which are in my story highlights on how to, some basic tools using Fiji, also called ImageJ, which is a free software by the NIH, and how to use this software in order to analyze your images and get data from them, and how to quantify your images. So we're not going to talk a lot about this today, but I just want you guys to know that this is out there. So once you perfect your protocols and get your images, you know what to do with them. All right, so this is going to be the outline of today's webinar. We're going to start off by going over the pros and cons of formalin-fixed paraffin embedded tissue called FFPE tissue. This is something that I kind of specialize in. Next, we'll be talking about what I think are the critical steps in immunofluorescence staining. We're specifically going to be talking about how to multiplex, so using multiple antibodies on a single sample. I'll go over some common problems and ways to troubleshoot. And lastly, I want to make sure that all your questions are answered as well. And before we start, I just want to say that everything we're going over today is just things that work for me. There are a lot of different ways to do immunofluorescence. And I think if you talk to a lot of people, you'll find that they each kind of have their little like tips and tricks that they like to do. So this isn't by no way like a comprehensive, like the only way to do immunofluorescence. This is just what I found has worked for me really well. And hopefully it'll help you as well. But I think talking to a lot of different people and seeing, how different people do different things would be helpful because you can adapt, what they do to your own work. All right, so we're going to first talk about the pros and cons of FFP tissue. So I think there are a few pros and cons to FFP tissue. And what I mean by FFP tissue is formalin-fixed paraffin embedded tissue. So this is tissue that is put into wax, paraffin, and is cut into really, really thin sections, usually 6 to 7 microns, and then mounted on a slide that is compatible for imaging. So there are a few pros to this. Because the tissue is cut so thinly, you can often get, tens, maybe even like 100 slides from a single piece of tissue. And this means that every single slide you can stain for something different. You can test out your antibodies because you have a lot of slides. So I really like this because, I can stain a single sample for many, many different things. As I mentioned, the sections are very, very thin, usually around 6 to 7 microns thin. And that means that this is really good for wide field microscopy because you don't have a really thick sample that gets like a lot of out of focus light. This is very, very good to do with wide field microscopy. Something else that I really like is that these samples can be stored at room temperature and often for years. So I keep all my slides and many of my blocks at room temperature. And I've worked with slides that are like 5, 10 years old that I'm still able to stain. And that's really great. This is in contrast to things like frozen sections, which you typically have to keep at like -20 or -80, and then typically has a much shorter shelf life. There are a few cons to FFP tissue. And the reason that it's stable for so long is because you've kind of processed the tissue a lot. And it's like, you've fixed it and then you've put it in wax and done all this stuff to it. So what ends up happening is there's a lot of antigen cross-linking. And this makes it actually the least compatible with antibodies. So typically you'll find antibodies that work for frozen sections or for whole mount. Or for Western blot, but there's like a good chance it actually might not be compatible with FFP tissue. And the other thing is that there's also a lot of steps involved from dissecting your tissue all the way to embedding and cutting, and then immunofluorescence after. So it's a little bit more involved. So that's kind of the trade-off between the pros and cons of FFP tissue. All right, so next we'll be talking about some of the critical steps in immunofluorescence staining. So here I'm just showing you a schematic of the general workflow for immunofluorescence staining. Usually you start with your tissue dissection and fixation, and we'll talk about this a little bit more later. Afterwards, after you've embedded the tissue and cut it, you'll put it through a process of deparaffinization, antigen retrieval, and blocking. And this is prior to immunofluorescence staining, where you apply your primary antibody and ultimately imaging and analysis. And we're really going to only be talking about the first 3 steps of these today. We're not really going to be talking about imaging analysis. We can save that for another webinar. So I want to quickly really go over tissue dissection and fixation. It's really, really important because, the quality of your tissue, if you're doing a dissection and you kind of like macerate the tissue, you're already kind of at a bad start. So you really want to make sure when you're doing your tissue dissection that you're really, really careful and you can preserve the integrity of the tissue. And I also want to say that immunofluorescence staining is really important and is kind of the starting line for every subsequent step. Every step like builds upon the step before. So you'll start with your immunofluorescence experiment and tissue processing, and then after you do your staining, you'll get your image acquisition, and then you'll do your data analysis, and then ultimately you'll have your actual data. And every I like to think of it kind of like a house where every floor, builds upon the floor below it. And you're having a really good immunofluorescence protocol and having really robust tissue processing is kind of the foundation. If you don't have a good protocol and your tissue isn't really good, then it doesn't really matter how good the rest of your stuff is. Your data ultimately might not be as like good or reliable or as repeatable. So it's really important. I want to emphasize that you have a good protocol and that you treat your tissue really well before you even do the rest of these steps. All right, so I talked a little bit about the importance of having a good tissue dissection and like making sure that you preserve the integrity of your tissue. Every single type of tissue will vary. So for example, I know for some people who work with bones, you have to put it through a process of like decalcification in adults. Some tissue contains a lot of blood. So some people like to do things like flush it out with PBS or flush it out with PFA to kind of fix it from the inside out as well as outside in. And I think that can be really, really helpful. So just take these things into consideration when you're processing your tissue. For me personally, I work mostly with embryos, which are pretty delicate, but I also don't have to do a lot of like processing or like the sort of decalcification that I think a lot of people with that work with like bones do. For example, if you work with brains, it might be a really good idea for you to perfuse your tissue with PFA before dissecting it out, because the brain can be really, really dense and it can sometimes take a while for the fixative to go from the outside in. So we talked about careful dissection and perfusion. I also want to talk about choice of fixative. There's a lot of different fixatives out there. What I personally like to use most is 4% PFA. You can even use something as low as 2% PFA for a little bit longer. And the reason I like PFA, which stands for paraformaldehyde, is that I find it's the most compatible with immunofluorescence staining as well as histology. There are other fixatives out there, such as 10% formalin, which is very, very common. The thing is that formalin contains methanol, which can often render your tissue incompatible with certain antibodies for immunofluorescence. So I prefer PFA for this reason. Other fixatives, such as Bowen's fixative, are particularly good for histology. If you want to do something like H&E staining or PAS staining, Bowen's fixative is excellent for that. However, that means that you probably won't get very good immunofluorescence staining because Bowen's fixative is notoriously bad and incompatible with immunofluorescence staining. So typically, in general, after I dissect a piece of tissue, I put it in 4% PFA. I actually probably put it for around 12 to 16 hours overnight at 4 degrees. You want to make sure you use a good amount of like excess PFA that the tissue can kind of like swim around in. And after that, at the PFA, what I like to do is a slow dehydration process. So after the PFA, I will put it in 1x PBS, and then I'll put it through a slow ethanol series of 50% to 70% to 100%. And each of these steps I do overnight at 4 degrees. I've seen a lot of other things out there. Like some people like to do PFA for a few hours at room temperature. And then other people like to do the ethanol for shorter amounts of time. This is just what I find works for me. If you're doing some things like RNA fish or potentially some other applications, I know that sometimes it's suggested that you cut from 4% to 2% PFA. And I think that's totally fine. I have done 2% PFA and I find that it's really very much compatible with immunofluorescence. All right, so we talked about tissue dissection and fixation. Next, we're going to talk about deparaffinization, antigen retrieval, and blocking. So here's the general workflow after you dissect and fix your tissue and put it through that series. You'll embed it in paraffin. Usually you'll do it, or a core will do it, or a technician will do it, and you'll cut it and mount it onto slides. And then after this, you'll put it through deparaffinization, often in xylene. So what this does, it removes the excess wax. And I really want to emphasize this portion. And I actually tell people that this is probably the most important part of immunofluorescence staining. And I think that's really surprising to a lot of people. I've seen a lot of protocols say something like you can just do like 3 changes for 2 to 3 minutes each. And just to let you guys know what I used to do is I actually used to do 3 changes of 15 minutes each. Now what I do is I actually just leave the slides in xylene for a minimum of 45 minutes, and I'll actually go all the way up to 2 hours. And if I'm not in a rush, I'll fully leave it in for 2 hours. The reason for this is that the purpose of xylene is to remove the excess wax. If you don't remove all that wax, it really doesn't matter your subsequent steps. You're not going to get very good binding of your antibody. You're not going to get good uncross-linking of your antigens. So I really want to emphasize how important it is to get rid of all that wax and to submerge it in xylene for long enough. I have found when I've kind of been in a rush and I try to do like 30 minutes that I usually don't get as good results. I know some people also like to put it in the oven and bake it a little before they put it in xylene. And I think that's totally fine and probably helps. Just in my experience, I haven't found too much of a difference between baking it and then putting it in xylene for an extended period of time and then just straight putting it in xylene for an extended period of time. So again, I really want to emphasize just how important this step is. I tell people it's probably the most important step of immunofluorescence. All right, so what I often regard as the second most important step is antigen retrieval. So the purpose of antigen retrieval is to uncross-link your antigens so that they are available for antibody binding. And there's a lot of different types of buffers used for antigen retrieval, and this includes sodium citrate buffer, tris-HCl, and proteinase K. What I like to do is sodium citrate buffer. And there's different methods of retrieval, particularly for heat, and that includes the microwave, the steamer, and the pressure cooker. Personally, I am a fan, as I mentioned, of sodium citrate buffer combined with the microwave, and you want to make sure that it's really in kind of like a large amount of buffer and it's continuously boiling. But I know a lot of people, particularly in my lab, have a lot of experience and success using the steamer as well. So again, this is, I think, the second most important step because you really want to make sure that you're uncross-linking those antigens so that they're available for antibody binding. And then next comes blocking. So the purpose of blocking is to prevent non-specific binding of your primary antibody. My preferred blocking solution is 10% normal donkey serum and 1% BSA that's diluted in PBS. And the reason for this is I find that donkey serum is most compatible with my antibodies. So some alternatives include goat serum or chicken serum. The thing is, if you use, for example, goat serum for blocking for your primary antibody, you can't use a goat primary antibody. Same with chicken serum. If you use chicken serum for blocking, you can't use a primary chicken antibody. I have never come across a primary donkey antibody. So I just find in general using donkey serum is great because I don't have to worry about whether it's compatible with the primary that I'm using. I see some people use BSA alone, and I think this is perfectly fine. I just think that you get a little more of a blocking effect and a little less non-specific binding when you add on donkey serum on top of that. There are also commercial blocking agents available out there. I don't really like using some of these agents, not because they don't work necessarily, but just because a lot of the recipes are private. They're not available for you to see. So it's really, really hard to troubleshoot if anything if anything doesn't work. All right, so next we're going to talk about multiplexing. And by multiplexing, I mean applying multiple antibodies to your sample. So in terms of our workflow, we're now here at the immunofluorescence staining, and there's a lot of different ways to multiplex, but before you start multiplexing, there's a few things you'll want to consider. And the first is the suitability of your antibody for your specific application, which is immunofluorescence. So there's a lot of antibodies out there, and it can be used a lot of different ways, such as, and immunofluorescence or immunohistochemistry, immunocytochemistry, or Western blotting, but not all antibodies are suitable for all applications. So oftentimes an antibody that might work for Western blot actually might not work for immunofluorescence. So you want to make sure that your antibody is suitable. And usually what I do is I look to see if this antibody has been published or has been used for the exact same application as I've done. So even for immunofluorescence, I prefer to see an antibody that has worked for paraffin tissue rather than frozen tissue. Next is species recognition. So you want to make sure that your antibody can recognize the species that you're working with. So if you're working with mouse tissue, you want to make sure you're not using an antibody that only recognizes the human antigen, because there might not be enough homology for cross-reactivity. Next is the quality of antibody and the specificity of antigen recognition. So some antibodies will bind to other kind of homologous proteins. So you just want to make sure the antibody you're working with is very high quality and that isn't going to bind to other proteins that are in the same family. And lastly, concentration of antibody. This is something we'll talk a little bit about later, particularly in terms of troubleshooting. So there are many ways to multiplex, and you can use both conjugated and unconjugated antibodies. We're first going to talk about unconjugated antibodies, and the general rule here is that you want to make sure that your unconjugated antibodies are different species. There is kind of like an exception that I'll talk about later, but in general, you want to make sure you're using different species. So here I'm just showing you, for example, if this is your sample, you'll have a rabbit antibody and a mouse antibody. You can apply those at the same time, which is why I've labeled 1 next to them. You can apply them to your sample at the same time. And after you wash it off, you can apply your secondary antibody that recognizes your primary antibody. And you just want to make sure that your secondary antibodies are in different wavelengths. So for example, here you would use an anti-rabbit antibody at 488 and an anti-mouse antibody at 568. And here's just an example of immunofluorescence staining using unconjugated antibodies of different species. So this is an adult mouse intestine stained for E-cadherin in yellow, which stains for the enterocytes, live 1, which stains for the lymphatics, and then CD31, which stains for endothelial cells. And you'll see here, this is an E-cadherin antibody from Protein Tech, and you get really nice demarcation of the different structures. And I added all 3 of these antibodies, since they are from different species, rabbit, goat, and rat, at the same time onto my sample. And then I added my secondary antibodies at the same time as well. So it's really a very quick process. So another way to multiplex includes using conjugated antibodies. And when you're using conjugated antibodies, this means the primary antibody already has the fluorophore attached to it. And when doing this, you can use antibodies of either same or different species. So this is really great because you can add it directly to your sample. The one drawback is obviously you can't control the fluorophore is already predetermined. So you can't decide on your own which channel what color spectrum it's going to be. So here's an example of a mouse cerebellum stained for beta-3 tubulin, and this is from Protein Tech that's conjugated to Corelite 488, and calbindin, which is conjugated to Corelite 594, also from Protein Tech. And you can see both of these antibodies are mouse antibodies, but I just added them at the same time to my sample. And here you can see you get really nice staining of the neuronal cell bodies as well as the neuronal fibers within your sample over here. And you get nice demarcation of these different structures. So this is really easy as well. Again, it's just a one-step process. You just add your antibody, even if they're the same species, to your sample at the same time. Another way when working with conjugated is to combine it with an unconjugated antibody. And in this case, you can have them be the same or different species. The main thing is that you want to make sure if they are the same species, that the order really, really matters. So for example, if you want to work with an unconjugated rabbit antibody and then a conjugated rabbit antibody, you would first apply your unconjugated rabbit antibody, wash it off, add your secondary antibody against your unconjugated primary, wash it off, and then lastly add your conjugated rapid antibody. And the key here is to make sure you have really robust wash steps in between. You want to make sure that you get all of the unbound antibody off before you add another before you add the next one. Because if you, for example, don't get all of this secondary off and then you add the primary, this secondary might also bind to your primary, your conjugated primary. So here's an example of combining conjugated and unconjugated antibodies if they're the same species. So this is a mouse embryonic spine that I've stained first with GFAP. So this is a mouse antibody from Protein Tech that marks astrocytes. So after I put the GFAP antibody, I washed it off and I added a anti-mouse antibody, anti-mouse secondary. And then after washing that off, I added my conjugated beta-3 tubulin antibody. And again, you can see there's really nice demarcation of structure. So in the center of the spine, this is a neuron-rich region. So it stains heavily for beta-3 tubulin. And you can see the interjecting astrocytes in the spinal cord. Here's another example of using conjugated and unconjugated. This is an adult mouse intestine. First, I stained with E-cadherin, and then I added my anti-rabbit secondary. And then lastly, I added, after washing all of that off, I added a conjugated cytokeratin. So you can see that this marks the apical part of enterocytes, whereas E-cadherin marks the cell-cell junctions. And then lastly, here's an example of combining conjugated and unconjugated antibodies, but this time with different species. So this is very simple. What you can do is you can add your unconjugated and your conjugated at the same time. And after washing it off, you can just add your secondary antibody to your unconjugated primary. So here I'm staining for an embryonic mouse heart, and I'm staining for desmin, which is an intermediate filament expressed in embryonic cardiomyocytes. PCNA, which is a marker of proliferating cells. And what you can see here is that all of these cardiomyocytes are actually proliferating. So this is a pretty unique feature of the embryonic mouse heart, that it's highly proliferative. And again, you can see you get really nice demarcation of structures. You get the PCNA staining where the nucleus is, which is what you would expect. Oops. And before we move on, I just want to briefly mention a new product from Protein Tech called Flexible, which is a way to conjugate your unconjugated primary antibodies. And Afrida will talk a little bit about this later on. I want to first say that I have not tested this kit yet, so I really don't have any experience with it, so I can't speak for myself about how well it works. But this is a way for you, if you're working with two unconjugated antibodies of the same species, that you can use them both on your sample. The thing I would caution you against is making sure you're using kind of an excess of your unconjugated primary with this linker, because any unbound linker that is not quenched by the quencher will end up cross-reacting with the other unconjugated primary antibody. So the main message here is to make sure you kind of use a little bit of excess of primary antibody so that you don't have as much of the unbound linker. But Afrida will talk about this a little later. All right, so now we're going to talk about some common problems and troubleshooting tips. So one thing I often get in my DMs on Instagram is someone will message me and say, my immunofluorescence didn't work. What do I do? And this is very hard for me because, obviously there's a lot of different steps and a lot of places where it can go wrong. So in general, what I tell people is, I tell them to check a few places where I think people commonly go wrong. The first is to make sure your reagents are all compatible with immunofluorescence. So this doesn't just mean antibody and that it's suitable for your application, but also your fixative reagent to make sure, again, I recommend PFA over something like Bowen's or 10% formalin, to make sure your reagents are all compatible with immunofluorescence. And I would say to also in terms of antigen retrieval, to make sure, I recommend doing sodium citrate buffer. And then lastly, you want to make sure after you dissect your tissue that you get it processed sooner rather than later. If you can do it almost immediately, like that's great, but you really don't want your tissue to be sitting in PFA or sitting in PBS, like at room temperature at 4 degrees or anything for an extended period of time, because that means the tissue will begin to degrade slowly. And that ultimately, when you embed it, that the antigens might all be degraded already. Another common question I get is I have a lot of red blood cell autofluorescence. So there are a few tips and tricks that you can do to fix this. The first is to make sure that the channel that's giving you the most trouble with red blood cell autofluorescence, to put it in far red. So that means either using like Psi5 or 647 spectrum, because I find that when you're in the far red region, you tend to get less red blood cell autofluorescence. Another potential fix is to switch imaging modality. So using confocal can be a good solution because it'll block out a lot of the out of focus light. And if all of this doesn't work, what I say is you can always just stain for red blood cells, and that'll help you demarcate between what is your true staining and what is like red blood cell non-specific staining. So for example, here I'm showing you staining for the bone marrow, which obviously contains a lot of hematopoietic cells. And you can see here, I'm staining for endomucin, which marks the blood vessels. And within these vessels, you can see these little round dots, and these are all red blood cells that have been stained non-specifically, either by endomucin or the secondary antibody. And what I've done on top of that here is overlaid staining with TER119. So TER119 stains red blood cells, and you can see now it's really easy to demarcate what is a red blood cell and what is true endothelium from the blood vessel. And here are a few products from Protein Tech that work really well for TER119 staining. So another common question I get is my antibody looks like it worked, but there's a lot of background signal. And to this, I say, I think when people encounter this issue, their first thing to do is they'll increase the concentration of their antibody. And I actually suggest you do the opposite, that you should dilute it more, because then this way you won't get as much non-specific binding, and whatever antibody is there will more likely go to your desired target. So if this is your issue, I recommend you diluting your antibody first. And the last thing I'll talk about is some people will say, oh, the structures look really, really blurry when I'm imaging. And I find this is more of an issue when you're doing immunocytochemistry, so staining cells and trying to look at subcellular structures. And for this, I would say the best thing to do is actually switch imaging modalities to something that'll give you a little bit better resolution, such as confocal or even super resolution microscopy can be really good at resolving those structures. All right, so that concludes everything that I've prepared for you. I'm really happy to answer any questions you have now as well.
Well, thank you so much, Derek, for that wonderful workshop. It was very informative. We have a lot of great questions, so we'll try to get through all of these. If we don't get through all of them, we will email you guys individually with responses to your questions. So we can go ahead and start with one that we got in the Q&A box. So Pascal says, my protein is poorly expressed in cells. Can I use an antibody at 1 to 25 dilution for immunofluorescence? Or what do you do for colocalization experiments?
Sure. So if your protein is poorly expressed, I think, if 1 to 25 works for you, I think that's totally fine. The dilution that works. I think using a higher concentration is better than something like amplification kits. I'm usually not a fan of that because it's really hard to control the amount of amplification. So I think 1 to 25 is perfectly fine as long as you're not getting like a ton of background staining or anything. So if it works for you, I would say go for it.
Great, thanks. So we have a question from Matthew, and Matthew asks, what are the impacts of fixing for a longer duration? For example, PFA for 3 days or 1+ weeks?
Yeah, that's a really great question. So I would really avoid doing fixation for more than 48 hours. what can happen is you can get excessive cross-linking of your antigens, and even with like really, really robust antigen retrieval, it can be hard to uncross-link that. And what ends up happening is like antibodies that might work usually won't work for your sample. So I would really suggest not going over that 48-hour threshold.
Katerina asks, can conjugated primary antibodies be used for other applications, for example, IHC, with secondary antibodies HRP conjugated?
Yes, definitely. So you can even use it for example, like Western blot. You basically treat it as a primary antibody. Of course, if you're using it for like Western blot, you want to make sure that like there's not going to be like overlapping if you're of a fluorescence spectra, but you can definitely use a conjugated primary antibody for something like IHC for with like bright field imaging.
Great. Hallie asks, do you put the slides in the microwave while it is actively boiling or in hot water straight out of the microwave?
Yeah, so what I do, you want to make sure that your buffer is boiling for as long of a time as possible. I find that people will often put it in a really kind of small container, and I actually put mine in a very big container of excess buffer. And you want to, this helps without like this helps with preventing boiling over. And what I'll do is I'll like turn it on for like a few minutes and then let it cool down, and then turn it on, cool it down, turn it on, cool it down. I'll do like 5 or 6 cycles of that just so that it's in that boiling buffer for as long as possible. And at the end, I take it out and I let it cool completely to room temperature. Usually takes about an hour. And I let it stay in that buffer. So I don't like put it into like hot water or anything.
So Katerina also asks, what do you think about Sudan black incubation to reduce autofluorescence?
So I actually don't have a lot of experience using Sudan black or like quenching with autofluorescence, reagents. Typically, I find that just like with good blocking, you'll prevent a lot of the non-specific binding. And if you combine it with good imaging techniques, you really don't have to do a lot of those like excess reagents to try and quench. So I don't have a lot of experience with it, but I find that blocking with donkey serum is usually good enough.
There's a question from Julie. Julie asks, I've read that you should use a serum to block that is from the same animal as your secondary antibody. So goat serum is using a goat secondary. So what
Yes,
are your thoughts on that?
Yes, that's true. So if for optimal blocking purposes and preventing non-specific binding, your serum should be the same animal as your secondary. So as I mentioned, I use donkey serum. So all my secondaries are donkey like anti-rabbit or donkey anti-mouse or whatever. Again, as I mentioned, I prefer donkey because I actually work with a good number of goat primary antibodies. So if you're using a goat primary, you can't use goat serum.
So there's, on a similar note, a question from Jonas where he says, concerning blocking, antibody should work the best in its own host serum, right? I thought goat antibody created by goat immunization should work the best in goat serum. What do you think? So it's kind of on a similar
Yeah,
line.
I don't believe that is correct. I think you should, because what happens is you'll actually get a lot of binding of your goat antibody to the serum itself rather than to the target. So you should really make sure that your serum is from a different species as your primary antibody.
There's a question from Agnieszka about, so it says, you stress the importance of using xylene as a deparaffinatation agent. Ooh, that's hard to say. What is your experience with xylene substitutes?
So I don't have a ton of experience with like other non-xylene substitutes. I think really any non-organic, like non-polar substance should work, but I think a lot of them can be very like harsh, like for example, like acetone or something. So our preference is with xylene. Actually, no, xylene is probably as harsh as acetone if we're being honest, but I've really only used xylenes.
Great. So we have a question from Will. I have found when using the microwave that my buffer tends to boil. Are there not negative effects of boiling the samples for extended periods?
For me, I find if you're using like the correct slides and make the sections are like well adhered to it, that you really won't get issues with like the sections falling off or anything. As I mentioned, I like to put it in an excess. So it's really below, the slides are sitting below where there's like a lot of bubbling, because I find that sometimes the kind of turbulence of the bubbling can kind of interfere and kind of degrade the tissue a little bit. So you really want to make sure it's sitting between that like boiling line. And the other thing is you want to make sure that your sections are covered by a buffer the entire time.
There's a question from Baptiste. Is there an explanation why a specific antibody might work for Western blot, but not for immunofluorescence?
Yeah, so the reason here is that when you are preparing samples for Western blot, usually you will like boil it, put it in a reducing agent. You're doing like, you're putting SDS on it, which kind of like coats your protein that's now unfolded in like a net negative charge so that you can run your Western blot. And so your protein is quite degraded, and this means that I shouldn't say degraded, unfolded, which means that there's a lot of availability for antibody binding. So in general, there is more availability for antibody binding in protein samples prepared for Western blot than in the native tissue.
There's a question from Seda. How can we detect false positive results for IF sections?
Yeah, very good question. So the key here is to make sure you have really good controls. So ideally, you can first test your antibody either with some sort of validation if it hasn't been validated. So this includes like, you could do like knockdown in vitro. The absolute best I would say is you can get like, for example, if you're staining mouse tissue, to get a knockout of that gene for that protein. And then to stain. So if you have a control like a wild type and a knockout, you should see that it should work in whatever tissue in the wild type, but that you'll see that there's no staining in the knockout. You can also do things like doing your primary without a secondary antibody, and then doing your secondary antibody without a primary antibody to see like what is like non-specific and autofluorescence and all of that.
Great. There's a question from CB. Do we need to go through antigen retrieval when fixing for short periods of time, such as 20 minutes?
So I would say for anything, if you're working with FFPE tissue, so it's embedded in paraffin, like antigen retrieval is almost a must. I find that like you will have much less success if you do not do antigen retrieval. If you're doing like frozen sections or whole mount staining, I don't do antigen retrieval for that. I find that the antigens are perfectly fine, and you can just go directly to staining.
There's a question from Kristen. Did you ever notice cross-reactivity with secondary antibodies and siRNA treated cells? And if so, why do you think that's the case?
So I'm not entirely clear about the question. Like, I don't know if it's regarding like the secondary binding to the siRNA reagent itself. I have stained siRNA treated cells, and I don't really see any sort of, like non-specific binding in that regard. So I would recommend checking your reagent. There could be some things like some siRNAs actually contain like a GFP or RFP like sequence with it as well, so you can identify which cells have taken up the siRNA. So I would just double check your reagent to make sure that it doesn't contain anything like that, because that might be what you're seeing.
Shreya asks, what magnification do you visualize your samples?
Yeah, so it depends. I would say a good general magnification is probably 20x if you're in terms of an objective, so that's 200 times magnification. I find that works for most things. If you really want good subcellular, structure visualization, I would recommend at least 40x. And then if you're kind of just want like a pan scan of the entire tissue, like 10x should also be fine.
Julie asks, have you ever conjugated the fluorophore to a primary yourself? Any kits that you can recommend?
So I don't have a lot of experience with this, as I mentioned, but I think Afrida will talk a little bit about this later.
Yeah, I'll talk a little bit more about our newly launched flexible kits that make antibody labeling really easy. It's very quick. It's a two-step process, and you have labeled antibodies within 10 minutes that you can use for staining. So we did that. So Harry asks, is there a typical percent blocking agent that you use for antibody incubations?
Sorry, can you repeat that again?
Yeah, is there a typical percent blocking agent that you use for antibody incubations?
Yeah, I find anything 5 to 10% serum works great.
So Magdalena says, is the fluorescence from conjugated primaries strong enough for detection? My understanding is that when using secondary antibodies, there's some amplification of signal.
Yeah, so your understanding is correct. You do get some amplification from using a secondary on unconjugated primary. I find it depends for a conjugated primary. So in general, I find that it's enough, especially if the conjugation is with like a good, robust fluorophore, such as like Coralite or Alexafluor. Like I find that usually you get very good staining. If you're working with like antibodies of different species, you can honestly always just add a secondary on top of the conjugated primary if you find that it's not amplified enough, if you're not getting enough signal.
So Vey asks, what is your preferred washing, elution, buffer, and conditions?
Yeah, I just use just regular PBS. I know there's some people that like PBST, which is also very, very good and perfectly fine. The reason I don't like the adding the T, the Triton X, is because I find that I kind of like working with like the surface tension. So when you're using some sort of detergent, it can kind of like spill over easily. So just for that reason alone, I prefer using PBS, but using PBST is totally okay as well.
There's a question from Amandine. Are there any downsides of doing antigen retrieval all the time? So even if it's not needed?
Well, if it's not needed, then you shouldn't need to do it. In terms of like tissue integrity, I have found like no issues in terms of like the integrity of your section or anything. I think it should be totally okay. But if you don't need to do, like there are some antibodies that work without antigen retrieval, so you don't have to do it, then you don't have to do it.
Yeah, then you have to decide like, do you want to try it without, or do you want to just do it every single time because you know it works?
Right.
So we have time for maybe 2 more questions. There's one from Rehab that says, how to detect the best antibody concentration to start with?
That's a great question. So I just usually will, if it's been published, I will typically do whatever is published. And I will also kind of often start with, sorry, that's my dog. I will often start like 1 to 200 or 1 to 300. That is my typical starting point.
And then, so let's end with this question. This is a question from Amandine. If we see blood cells in our tissue, is this a sign that perfusion didn't go well, or is this quite normal actually?
So it's not necessarily an indication that perfusion didn't go well. I think perfusion definitely helps, but vessels are very, very tricky. And then there's like, all different types of flow. Sometimes red blood cells can be sticky. So I think, excessive perfusion helps, but doesn't necessarily mean that your perfusion didn't go well.
Great. Okay, well, thank you so much. There are several questions that we didn't get to because of time, but like I said, we will make sure that we email you guys individually, and Derek will answer your questions. I'll try to answer any of the questions that are directed towards protein tech, and we'll definitely get that over to you guys. With the last few minutes that we have, I do want to share just a few more slides. And I just want to share a couple products that I think might be beneficial for people that are doing IF, multiplex IF experiments. Before I do that, I do want to share our contact information. So if you want to directly contact Derek with any questions, you can email him at dereksung512@gmail.com. Or if you have any questions for me, you can email me directly as well at afrida@ptglab.com. So with that, the first product that I really want to highlight are our correlate conjugated antibodies. So Protein Tech offers primary antibodies that are conjugated to our correlate dyes. And our correlate dyes are synthetic dyes that are equivalent to Alexa Fluor in their spectral profile, their brightness, and stability. And we offer conjugated primary antibodies in correlate 488, 594, and 647. And all our conjugated antibodies undergo stringent validation so that we can obtain reliable results. So if you look here on the right, this image over here, this is actually an image that we obtained from Derek, where he stained using correlate 488 CD31 and correlate 594 alpha tubulin in endothelial cells. And you can see that we get very crisp images with very low background. Another product that I wanted to highlight, which we have talked about a little today, are our flexible antibody labeling kits. So this is a brand new product that we just recently launched. And Flexible is a novel antibody labeling kit that uses an affinity linker to conjugate fluorochromes, enzymes, and molecules in any buffer condition. And what makes this kit so great is that it's compatible with primary antibodies from any supplier. So it doesn't have to be a Protein Tech primary antibody. You can use your favorite antibody from any other company and still use our flexible kits in order to label it. It also works with any antibody concentration and formulation. So you also don't have to worry about what buffer your antibody is coming in or what concentration the company has provided for you. Flexible is also great if you're just trying to label enough antibody for 1 or 2 experiments. So you can label as little as 0.5 micrograms, and there's no buffer exchange required. The protocol is also very fast and easy. It's only about 2 steps. So your labeled antibody is ready to go and be used in your experiment within 10 minutes. And with one of our standard kits, you can actually label 50 different antibodies and no additional equipment is required. So everything that you need in order to label your antibody is going to be provided for you in the kit. So as I mentioned, this is a newly launched product. So right now we have flexible kits that are available for labeling of rabbit IgG and mouse IgG1 antibodies with correlate plus dyes. However, we do have more in the pipeline, so do stay tuned. I also want to talk a little bit about how our flexible kits work. So the way that this would work is you would have your primary antibody that you introduce to our flex linker, which it has the conjugate attached to it. And so basically what happens is your antibody binds to the non-covalent flex linker, and the flex linker will bind very tightly within minutes of being put into solution. And so then what you do is you neutralize the unbound flex linker by adding a flex quencher. And so what you're left with then is a homogeneous, well-defined antibody that's fully functional. And when we've used these kits, we haven't observed any aggregation or loss of antibody after labeling. So the flexible kits are ideal if you're trying to multiplex. And so I wanted to show 2 examples of how the kits work while multiplexing. So for example, if you're trying to multiplex with antibodies from different species, what you can do is you can take your primary antibody and mix the antibodies and the flex linkers together in the same tube at the same time. And so basically what that means is that your antibodies are ready to go within 10 minutes and you already have your multiplex mixture that you can then apply to your sample. So on the right, what we're showing is an example of this. So we have calbindin labeled with flexible correlate plus 550, ACE2 labeled with flexible correlate plus 650 kit, and correlate 488 conjugated podocollaxin, and this is in kidney tissue. And you can see that we get very crisp and very distinct staining utilizing these flexible kits. You can also use the flexible kits if you're trying to multiplex antibodies from the same species. So if you have several antibodies from the same species, what you would do is you would take your antibody and then label them individually with the flex linker, and then mix all the label antibodies together, and then apply those to your sample. And so here on the right, that's exactly what we did. There's 3 different antibodies here that were from the same species that we labeled using our flexible kits. So we have TDB43 labeled with flexible correlite 488, TOM20 labeled with flexible correlite plus 550, and lamin B1 labeled with flexible correlite plus 650. And this is in HeLa cells. And again, you can see that there's very crisp and very distinct staining of these proteins. And then lastly, I just want to point out our nanosecondaries. So our nanosecondaries are secondary antibodies that bind to primary IgG antibodies in a subclass specific manner. So basically, our nanobodies are conjugated to Alexa fluor dyes, and they are manufactured recombinantly, and they have a defined DOL. And these nanosecondaries are especially useful if you're trying to use this for confocal or super resolution microscopy, as well as multiplexing. And there's 3 main benefits for our nanosecondaries. The first is the faster staining protocol. So nanosecondaries are monovalent, so they don't cluster. So what this means is that you can simultaneously incubate your primary antibody with the secondary antibody and save time in your staining protocol. So this basically cuts your staining protocol in half because, with your traditional sequential staining, you have your primary incubation, your washes, and then your secondary incubation. But with the nanosecondaries, you can just use you can just do the primary and nanosecondaries together in one step. The second great thing about the nanosecondaries is the higher resolution imaging. So nanosecondaries are very small in size. So what this means is that at the end of the day, the nanosecondaries are closer to your protein of interest. So you have a small epitope label displacement, which leads to a higher resolution image, as you can see here at the bottom. And then lastly, we also see very clean images with our nanosecondaries. So our nanosecondaries are subclass specific and they generate very low background, which is ideal for multiplexing. So here in this plot, you can see we tested our nanosecondaries, and each nanosecondary recognizes the subclass, the IgG, that it is supposed to. However, when we looked at a competitor secondary, we see that it does recognize rabbit IgG, but it also recognized the IgG of different subclasses as well. So those were just a few products that I wanted to highlight that I thought might be helpful for anyone who is trying to do a multiplex IF experiment. I also wanted to let you guys know that as an attendee of today's webinar, you are eligible to receive a free sample of any protein tech antibody, and this includes our correlite conjugates and our chromatech nanosecondaries that I talked about today. And we'll provide this link for you in a follow-up email, so you can just click on it and fill out the form and request any free sample of any protein tech antibody. So with that, I will go ahead and end the workshop. I want to thank Derek once again for leading such a great workshop, and for the audience for having such wonderful questions. We will definitely get back to you guys if we didn't get to answer your questions live, and hopefully everyone has a great rest of your day. Thank you so much.